Continuing with our discussion of MRI modalities, we have quantitative susceptibility mapping, or QSM. This is a relatively new modality that's starting to be used more broadly now. The appearance is a grayscale image where the intensity corresponds to the concentration of specific particles. So we can use this to quantify certain things of the brain like iron and calcium. One example application is in this study with Parkinson's disease. They found that patients with Parkinson's showed iron deposition in the caudate, which is this area right here on either side of the brain, particularly in those who are showing signs of mild cognitive impairment. Iron can be neurotoxic to the brain, meaning it can be toxic to the neurons and thus lead to tissue damage. Next modality is arterial spin labeling. This also shows up like a heat map, kind of similar to fMRI. Again, similar to fMRI, it measures cerebral blood flow. It measures tissue perfusion. So it does this in a different way from fMRI, but you can kind of conceptualize these as similar in that regard. One example application of ASL. ASL can show alterations in migraine. So in this study here, they have two participants who were having a migraine during their scan compared to after when they were asymptomatic and had a follow-up scan. And you can see certain brain regions are hyperperfused when a migraine is occurring. So this supports the idea that there is a vascular component to migraine attacks. And in some cases, ASL can detect some changes right before the migraine occurs. So this can tell us a little bit more about how these headaches emerge. Next modality is magnetic resonance spectroscopy. This one is a little bit different in that what we get from this is a spectrum, generally, rather than an image. You can get a whole brain image from this, but you get this spectrum. You can get multiple spectra from multiple different regions. And in this case, the voxels are usually quite large, like 4 millimeters cubed. And you get a spectrum that can tell you the levels of various metabolites in the brain. So from this spectrum here, some of the peaks that we can look at are NAA, N-acetyl aspartate, which is a marker of neuronal health. Choline, which is a membrane marker. It's a marker of gliosis, which is like scars in the brain, cellular turnover, and inflammation. Creatine is an energy marker, so if this is low, that can be an indicator of cellular dysfunction. Myoinositol is a glial marker. These are the cells that respond in inflammation, and so higher myoinositol can be an indicator of inflammation in the brain. The glutamate-glutamine ratio, this can be an indicator of neuronal death. And lastly, lactate, which is not shown on this particular spectrum. Lactate is a marker of anaerobic metabolism, and it can indicate diffuse axonal injury. This is something that occurs in traumatic brain injury where axons are stretched, in some cases to the point of breaking, but even if they don't break, they can be stretched, which can offset the balance of the inside the cell versus outside the cell, and thus cause diffuse axonal injury. MRS example application. In this case, we're looking at a patient who had a stroke, and we can use MRS to compare the damaged tissue to the healthy tissue. So for example, here you can see the spectrum from the healthy tissue here, looking at the NAA peak. And then in the damaged tissue, this is the spectrum that comes out where we see a much slower NAA peak. So clearly neuronal health is impacted as we would expect. And lactate peak. So there could be anaerobic metabolism occurring here. So we can use this here in this example. The patient can be their own control. You can compare the damaged area to the healthy area. And we can use this to track how tissue recovers over the time, or in some cases doesn't recover over time. Positron emission tomography. This one, generally what we look at is a heat map like this. And the way that this one works is a person who's undergoing a PET scan is injected with a radioactive dye. It's a very small dose of radiation, but a radioactive dye that can bind to different types of cells depending on what tracer is used. So one of the ones that's used most often is FDG, or fluorodeoxyglucose, which is a glucose compound, so it can show metabolism. So if you see peaks, hotspots in a PET study using FDG, those hotspots would be places that are more active, thus their metabolism is higher. Another one that is used quite often is fluorobeta-PIR. This one binds to amyloid, which is of particular interest in Alzheimer's research. An example application here from Alzheimer's disease, using a different compound that binds to amyloid, Pittsburgh compound B, or PIB. This shows where amyloid has accumulated in Alzheimer's disease, which is pretty extensive compared to a healthy control. Single photon emission computerized tomography, SPECT. This one shows up as a heat map, and this detects how well blood flows through the brain. There's a radioisotope that's attached to a particular ligand that binds to tissue. So it's similar to PET, but the radioisotope that they use lasts longer and is less expensive. It also emits a different type of array, a gamma ray, which are picked up by a gamma ray camera, which is also relatively inexpensive compared to PET imaging. One example application here, looking at depression, and these top images, these show for one individual patient hypoperfusion, meaning underperfusion of the tissue in this individual who has been resistant to treatment for their depression, meaning they haven't been responding to the medications they've been on. And after a new treatment is tested, we see increased perfusion. So in this case, this is a good thing. And looking behaviorally as well, looking at symptoms, this increased perfusion was correlated with increases, with improvements in behavior, with improvements in depression symptoms. Computed tomography. In this one, low density tissues such as fluid appear dark, and high density things like bone appear light. This is usually used to find acute pathology like bleeds in the brain. And here, this example application is actually multiple different TBI patients showing the different types of neuropathology that are visible on a CT scan after traumatic brain injury. So we have epidural and subdural hematomas here. We can see contusions, which are bruises in the brain, subarachnoid hemorrhages. These hemorrhages and hematomas are all bleeds in the brain, and diffuse swelling. These are all visible on a CT scan. Electroencephalography, or EEG. With this, participants have a cap of electrodes on their scalp. You don't have to shave your head. They have a conductive material that is placed underneath them, so they just stick it to your scalp through your hair. And you can measure surface level brain activity through the electrical activity. So each one of these electrodes gives a reading such as this one here, and you get this around the brain. One example application is in epilepsy. This can show what seizures look like in the brain. So this down here is during a seizure compared to healthy. But this can also detect differences in between seizure episodes and right before a seizure. And lastly, magnetoencephalography, or MEG. Again, like a heat map is the appearance. And this one is a little bit different from the other ones in that you sit in this gigantic helmet of sorts, and that's what measures the activity. MEG measures fluctuations in brain activity through the magnetic fields that are created by the electrical currents in the brain. So whereas EEG is detecting the electrical activity, this one is detecting the magnetic fields that result from the electrical activity. And there are multiple different frequency bands that can be extracted from MEG readings. So what a frequency is just how quickly something is varying. So you can have slow frequency oscillations, you can have high frequency oscillations, and everything in between. And each of these can be extracted separately and can tell you something different about brain activity. So there are several different types of frequency bands that are commonly extracted. And one example application here, again in traumatic brain injury, they found differences in the frontal cortex in individuals who had had a TBI with a loss of consciousness compared to those who had a traumatic brain injury but did not lose consciousness. So in this case, lower oscillations, lower activity, lower energy. And that is it for our very, very basic introduction to neuroimaging. We will go into a lot more detail in the future.